It's a small thing, it's like 3 days. Yeah, nice. But yeah, they're literally. Mm-hmm. There's a lot going on in that field right now. There's a help part.
All right. Hi, everyone. My name's Tracy McGinnity. I work in shared resources here at Wilmot. Today, we have our Biobank Shared Resource Seminar. I'd like to start by highlighting our support is from Vivian Palladoro and her endowment. Vivian's a retired URMC clinical pathology administrator, and she provides the funds to help us get the word out about new services the shared resources may be providing, and highlighting the expertise of our shared resource staff. So she has been putting this on. This is the second year we've offered this series. And we also have the upcoming schedule. So today's our biobank. In October, we have our metabolomics shared resource presenting on untargeted metabolomics. And then in November, you'll hear from our biostatistics and bioinformatics shared resource. December, I think, will be a community event. If you're taking part in the communicating science workshop, we are trying to organize that as an opportunity for you to share your research kind of in the realm and through the lens of shared resources, so highlighting the different services and equipment that you utilize for your research. Other events that we have upcoming on Friday, October 17th, we have our CCAC retreat. This is aimed at presenting your research for community members, and they come and give their feedback of how they'd like to interact with the greater research that's being performed here at Wilmot for cancer. And on Thursday, November 6th, we have our Wilmot Symposium. And then finally, we are creating a 2026 shared resource calendar. So if throughout the year you created a really impactful figure or image for a paper or utilizing your shared resource, we would like submissions to be entered by Friday, September 19th. This year, we're trying to highlight those publications that were really impactful for us getting our NCI designation. So that's kind of our brief interview, and then I will let Brad take it over from here. Oh. Was you? Yeah.
Thank you, Tracy. Thank you, everyone, for attending. Pretty good turnout. I'm happy with it, and I hope you get something out of today. So what I'm going to go through today, based off of registration feedback, I'm going to give a brief background about the biobank, the services we offer. I'll go at hyperspeed through that, right? So the pace will start out quickly, just to touch on all of that. As I get into sort of our collection and processing workflows, I'll slow down a bit because I want to highlight, I think what it will do is highlight some of the tissues that are available, some of the derivatives we can make from those tissues and how they're collected at different time points. And you can begin thinking about how those can be adopted into different preclinical models. And then, where I'll really take some time, hopefully, is in patient-derived tissues for ex vivo modeling. What I'm going to focus on primarily are culture models. I'll touch upon PDX, some engineering-based methods, but I'm really going to focus on tumor-based patient-derived culture models for that. So here's where you can get to our website off of the Wilmot research page by clicking on Biobank Biospecimen. It'll take you to our sub page. Everything you need to know is on there, how to contact us, and absolutely feel free to stop by. So we're just off the yellow elevators on the 6th floor. Unfortunately, our hallway is Vivarium access, but there's signage everywhere. Give us a call. We're there all the time, so please feel free. So what do we offer? we're a biobank. At the heart of what we do is collect biospecimens and develop this retrospective inventory of different samples and corollary clinical data used to annotate them. Beyond that, though, oh no, sneak peek. Beyond that, we also, of course, we offer advanced data. These patients are enrolled through informed consent, so we can pull data and provide de-identified data across the board. And also we provide tailored study support, and that's getting into prospective collection and study-specific collection, which I'll talk about later. And with that tailored support, we offer much more. So project design, developing methods or models, and even getting your sub-study on our biobank tissues approved through the RSRB. And lastly, a quick plug for the expansion of our spatial omics suite. So as I'm sure all of you know, our GRC has Xenium, Visium, NanoString, spatial transcriptomics platforms. We have funding now to augment these with multiplex immunofluorescence. So next calendar year, we'll have a Lunaphore Comet here. It's basically a super fancy microfluidic system with a fantastic camera on it. So your favorite primaries, you'll stain them in cycles of 2 primaries with a fluorescent secondary, stain, elute in a cyclic fashion, and you can build 20-plex protein panels pretty cost effectively. So as I mentioned, our retrospective inventory is in our freezers on the 6th floor. You can access this at any time by reaching out. And we prospectively collect these tissues with Wilmot priorities in mind, also being mindful of what research is going on at the institution and prevalence of cancer in our catchment area. So of course, we have a good number of lung and breast tissues, prostate, colon, but pretty diverse cohort across the board. And we can expand and contract this. So even if you're interested in a tissue that's prevalent in the region, being looked into by several investigators here, we can start collecting these without a specific project in mind. So feel free to reach out. And we have done so. As you can see, we've really expanded our breast collection over the past few years, as well as started bringing in more cases of bladder. When we collect blindly, if you will, for this retrospective inventory, generally we're doing more generic preservations, a lot of FFPs whenever possible in blood derivatives. But our SNAP frozen inventory now includes some viably frozen tissues to support the modeling I'm going to talk about. And really, FFPs especially are invaluable, both for the spatial applications I just mentioned, but as well as validating these models, which I'll get into a bit later. Reach out to us. So we have a partition of our database that's accessible to all university investigators. So we can give you access to this. It lists the cases we've accrued, and then under another tab, it lists all the samples we have for those cases. So you can find not just tumor FFP blocks, but matched blood specimens. And from there, after we talk about what you may be interested in, we have templates and provide support for getting your project approved by the RSRB. So even though you're using de-identified samples, they still like a submission. Typically, it's exempt from human subject research. And then beyond that, we have a quick REDCap submission process. We have a utilization committee that oversees who's using these specimens and ensures that they're being used the right way before we disperse them. But it's quick and easy, and we're more than happy to help you through it. So now to start getting in, that was our retrospective inventory. To think about study-specific or prospective collections, right? Collecting tissues for specific projects, it really starts with the protocol we have in place to collect these tissues. So we've just drafted a more universal banking protocol to allow us to be agile. So we don't have to put in a separate study to collect a unique biospecimen or something we haven't already been collecting. What we tried to build into this through informed consent is obviously the ability to collect surgical resects, but beyond that to collect other samples from that patient longitudinally through recontact. So the patients are consenting for us not to just collect on the day of their surgery, but perhaps at infusions later on, or to ask them for a urine sample along the line, or serial urine samples, both for biomarker studies, but as well, especially with blood, for reconstituting some of these models that I'll talk about later. We've also written in to expand this process, doing this at offsite locations, right? So, for example, breast cancer blood at Pluta, or satellite infusion clinics. We can collect samples from those as well. In consent flexibility, I won't get into, but essentially what I'm saying there is we have more flexibility as to how we enroll these patients for really seamless onboarding of these different cohorts. And we're also in the process of drafting a universal pediatric protocol along these same lines. And we're looking to build both a retrospective pediatric tissue bank as well as support prospective research in the same manner. So this got super grainy, but that's okay. So to kind of take you through prospective collection, right? It's the same process for which we build our retrospective inventory. But by taking you through this process, what I want to highlight is kind of when we get these tissues, what we're doing with them, and to give you an idea of how we can collect different tissues or even different fluids and process them in different ways to support modeling, right? Not all of these are applicable in a modeling scenario, but a lot of these, I think you hopefully by the end of this talk will be able to see how you can adopt these into a model or use them to study specific mechanisms or settings of malignancy, right? So of course, we'll start with the foundation, right? Resected tissues. Whenever possible, we're not just collecting tumor, but we're collecting the invasive margin as well, which at times gives you, almost completely normal tissue. There are occasions in certain cancers where we can get contralateral normal, which is a much better control. But, really the tumor, in most cases, we're at least getting tumor, and that's what we're looking for. So as I mentioned, FFP is, as generic as you come, but a very powerful and useful preservation. Maybe second in line to that is SNAP frozen. Fresh tissues are really what we're looking for the modeling systems I'll talk about later. But in addition to that, and to build some level of retrospective inventory that you could tap into any time, we're starting to viably preserve some of these tissues as well. So how are we doing that? And really, what are you getting there? Going from fresh to, post-thaw from a viable preservation, you are going to lose granulocytes, right? If you're looking at neutrophil biology, you need to work with fresh tissues. But beyond that, aside from perhaps more necrosis in some settings, you're getting viable fragments that absolutely can be used to model, and many groups use these preserved tissues to model from. There, really isn't anything super sophisticated about preservation. Many reports have shown, and most groups tend to freeze these in small, like 1 to 3 millimeter cubed tissue fragments rather than single cell dissociations. They tend to preserve better that way, but also they'll support different processing for the models I'll talk about later. I think that'll come into view. But beyond that, it's either proprietary or a standard slow freeze media, right? CryoStor is an option used by many, a proprietary media, but simply FBS and DMSO in a controlled freeze will produce viable fragments downstream. And I think this is a great example of why it's good to work with the biobank to procure these tissues. and to process them and store them for these downstream assays, especially preclinical modeling. So what I'm showing you here is an example of one of our dissection images. So this, I believe this was an adrenal tumor, relatively small piece that we got from surgical pathology. But what I'm not showing here is the gross image of that tissue where it came from, right? So even with respect to gross anatomy, we're tracking to the best we can in the time we have where this tissue we got for banking came from, right? From there, what we're denoting with these boxes are how we dissected it further and what we did with each of those specimens, right? So on the left, this piece was snap frozen. In the middle, we took an FFPE, and on the right is the tissue that we further digested into small tissue fragments for viable freezing. In our inventory management system, we have a pretty robust setup where we can track the processing of these tissues, right, and the further dissection. So what you can see here from this case, right, and this is one collection. If we had serial longitudinal collections, we would have another branch coming out, right? So we're tracking all these derivatives back to the patient. But from this large piece of tissue, or the greater piece of tissue we got from pathology, we see here we have these three sections, right? A snap freeze, viable tissue fragment, which was put into 3 different vials in this FFPE. Where this comes into play and why it's so important is you can see here, we track QC, but the block orientation, right? So what we have listed here, the face of the FFPE block relative to the image we see here is the right side, and the top of that block is the upper portion. So as you can imagine, that piece in the middle was turned like this and put in an FFPE block. Why that's important? What that tells us is if we take a 5-micron section off the top of that block, that's the region of tumor that's close. It's immediately adjacent to the slow frozen sections. So if we're validating our model, and I'll show later, you really want to validate, most importantly, to the in situ tumor, right? To that fresh piece of tumor. We can do that by noting how we oriented that FFPE block and what came from in the greater tissue relative to those slow frozen sections. So it may seem boring, often goes overlooked, but it's super important when you're really looking at variable biology in the human setting and trying to model the entire 3D reconstruction. So in going through this process, there are a couple of works I want to highlight for different reasons. These aren't modeling studies that were done. They're more characterization and in more common uses of a biobank inventory, right? Sampling different time points or tissues, from a cancer setting. But this in particular, this study of tumor interstitial fluid, I want to highlight because I think it gets us thinking about the tumor microenvironment that we're trying to capture in our preclinical models, right? So tumor interstitial fluid is a very simple and easy to isolate derivative. We're essentially centrifuging tumor tissue on a filter at slow speeds. So we're not rupturing cells, but we're isolating all of the fluid that's inside that tumor. So really easy to do, but very much understudied, right? We always study the tumor cells and even stromal cells in the TME, but less is done studying the acellular physical stroma, and even far less is done studying the fluid that exists in that space between cellular and acellular stroma. So a lot of this work as of late has been driven by Matt Vanderheiden and Alex Muir out of University of Chicago. And this is one of the profiles they did in renal cell carcinoma. So it was super simple. They took fresh tissue from kidney resections, isolated tumor interstitial fluid, as well as kidney interstitial fluid, or KIF, from adjacent normal kidney, as well as plasma, and performed polar metabolomics as well as lipidomics. And what this shows is kind of contrary to especially for metabolic researchers, their I think traditional belief in the cancer microenvironment. What it shows, looking at these PCA plots, is that plasma metabolites are unique, and they separate from what we see in interstitial fluid. But tumor interstitial fluid doesn't separate, especially on the lipid level, nearly as much from normal as we once thought, right? These tumor cells are aberrant, metabolically aberrant. They're producing, they have very high metabolisms and are producing and excreting a very different metabolite profile than their normal counterparts. But when we look at it here, there's not that much difference, right? So I think in thinking about modeling, especially in the tumor microenvironment, right? It's not just about what cells are in there. That's the starting place, right? But it's about the support structure of those cells and also the metabolite in nutrient bioavailability to those cells. And please stop me at any time to talk more or less about any of these things, any of these concepts. So thinking about, serial collections, longitudinal tissue is a possibility there. There are in certain cancers re-resections, metastatic resections, biopsies, but really the tumor tissue we're getting from that primary resection is the most reliable, right? I think when you're talking about serial collections, as I mentioned, we can collect saliva, urine, stool, all very interesting, but blood, is the mainstay there. But also it is the most useful when it comes to modeling, reconstituting those models and even deriving them because of all the constituents we can pull from blood. So circulating tumor cells, I'll mention later, we can build culture models off of circulating tumor cells. It's not the most common. It's being done more and more. I think this is a very interesting avenue. But beyond that, reconstituting some of the models I'll talk about with your immune populations, if you're not maintaining native stroma, and also treating them with extracellular vesicles or plasma derivatives, right, to see how they respond in that culture model. One study I'd like to highlight here, again, a characterization, but really a great use of multiple tissues from these patients in different types of tissue and different derivatives of those tissues. So this is work by David Braun in clear cell renal cell carcinoma. Again, I'm not biased. It just so happens I picked these two. But what this paper, what they did in this work, and again, grainy, sorry about that, is develop a personalized neoantigen vaccine for renal cell carcinoma. So to develop this vaccine, they actually started with fresh tumor resects from the kidney, performed whole exome sequencing, and predicted neoantigens. And from those neoantigen predictions, they engineered synthetic peptides to vaccinate these patients against their specific tumors, right? After vaccination, they collected blood serially over the course of the therapy, so priming and subsequent boosts. And through those blood collections, they really monitored patient responses, right? So they performed TCR sequencing and looked at the T cell repertoire that was specific clones to vaccine neoantigens, tumor, as well as a viral antigen. Also with this blood and with the banked tumor tissue they had, they homogenized those tumors into lysates and performed a interferon gamma ELISpot assay, right, to see if blood cells from those patients after vaccination responded to tumor antigen. And in fact, that's what they saw. So again, I think this is a great example of different ways you can take samplings from different sites from a patient donor and perform some really elegant studies, but none of this has to do with modeling. So let's get into modeling. These are purely characterization approaches, which are useful, but we're here to talk about modeling. In general, this is very timely given the special emphasis from the NCI or the NIH on developing more robust models, more robust preclinical models, both to provide alternative sources to a lot of the mouse modeling that's being done, but to also hopefully improve translation efficacy of a lot of the therapies we're developing. There's a big gap between some mouse models and what the patient histology truly is. So this is an attempt to start to get around, improve upon that, we'll say. So just, a very general overview to make sure we're speaking the same language. There's a lot of terminology out there. We'll go over, I just want to, mention some of these different preclinical models, right? So of course, we all know two-dimensional cell lines, low cost, highly reproducible, but where they lack is the biological relevance to the human condition. Stable cell lines are going to drift. You don't have a 3D microenvironment. You can do cultures, but again, you're in 2D culture. It's very cost effective, generally easy, but not what we're looking for with respect to the human condition. Maybe the next step up would be a spheroid culture. So this is where you're not growing two-dimensional adherent cells, but you're populating tumor cells from patient-derived tissues as spheres. This generally is a better representation of the human setting. It can show interpatient variability. It's more reproducible, but again, you're losing stromal components of those tumors, and there are also culturing issues with those. You tend to get a bit more drift. You're not seeing the varying clonality that you see in human tumors. And because in a lot of that is because of the spheroid condition where you're getting anoxia at the core of these spheres, cells are dying, and there is some selection occurring. The next step up from that, I would say, is an organoid culture, right? Where instead of floating spheroids, you're growing organoids. Oftentimes they appear as spheroids in a matrix, right? So that allows these cells to migrate a bit, to form three-dimensional structures. And these organoids can also maintain either native or reconstituted stromal populations, fibroblasts, immune cells as well, depending on the culture conditions. With those gains, you're also seeing a higher cost. You're seeing a variability in how well they recapitulate the tumor microenvironment and how much they cost to get that done, right? I think there's a pretty direct relationship there. But again, a step up and far more accurate than spheroid or obviously 2D cell lines. I won't get into much today, xenografts or microfluidic chips. Again, with both of these, you're talking higher costs. With rodent xenografts, you have a true physiologic system. You're propagating this tissue in, but unless you're spending even more money for humanized mouse models, you're losing the immune component in selection of those tumor cells based on the immune component. And with microfluidics, I think there's some really nice channel setups. Jim McGrath is giving a talk this Thursday, which I think will be heavy on organ on a chip, and it'd be great to attend. But that is slightly different direction from the culture models I'm going to talk about right now. So in thinking about this, right, we have ex vivo culture models derived from various organs. A lot of work has been done here. What I'm going to focus on are again, narrowing it down to tumor models derived from tumor tissue, keeping in mind that there are other ways to derive these tumor organoids. So from normal tissue, you can develop normal organoids and then perform gene editing to make these malignant in nature. If you're studying origins of cancer or functional mechanism of tumorigenesis, this is the way to go. Rather than going from normal tissue, you can also take induced pluripotent stem cells and similarly generate a normal organoid and go from there. But I'm focusing on tumor and tumor-derived, not circulating tumor cell-derived, which is the one other option. And, as I mentioned, and I'll get into further, These organoids can have a native stroma that you try to carry from the tumor, or you can reconstitute it through immune cells, or use these immune cells for co-culture with a native stroma. So there are a few options there. So building complexity into these models. So when you think of a tumor-based organoid model, as I mentioned, there are these two, I would say, main branches. You can reconstitute a lot of that stroma after a total digestion of the tumor tissue, or you can try to maintain the native architecture. And there are pros and cons to each of these approaches. I think just generally looking across, the main variable here is the level of manipulation and perturbation of that native tumor you're carrying out. So if you go into a complete dissociation in a single cell suspension, you're going to completely lose the 3D architecture, but you're going to end up with a model that's more reproducible and a model that you can carry for a while in culture. Partially digested to no enzymatic digestion at all and just mincing, then you're ending up with more intact native elements, both cellular and acellular. But with that, there's much more variability because you're closer to the human context, and you can't culture them nearly as long because similar to spheroids, they have to be cultured in a way that they're going to get sufficient oxygen and nutrients, or the culture is just going to drop out. So I would say it's a bit of a balance as to what model you select. And that's the reason why I'm presenting all of these. So you can see what may or may not apply to the specific mechanism you're studying. So to start first, the reconstituted side of things, single cell dissociation of these tumors. And these are typically grown, in each of these models I'm discussing are grown in a collagen matrix, right? Now, in co-culture models, you could have a suspension culture of, immune cells, right? Or fibroblasts, even though they probably prefer monolayer, but they can be mixed culture models as well. But the tumor organoids I'm talking about are grown in, these little matrigel domes. And on this side of things, you're typically submerging those domes in culture media. Because these are tumor organoids, they love hypoxia. They can grow in those conditions. They'll proliferate. There'll be, I would say, some level of selection and drift to a tumor cell that's, equipped to handle those conditions. But in the paper I'll show coming up, they much more accurately recapitulate the human tumor histology and type than a 2D cell line or even a spheroid for that matter. So when it comes to reconstituting your CAFs, your fibroblasts, your immune cells, as I mentioned, the way to do that would be a co-culture with, primary isolated cells or with genetically engineered cells or primary cell lines, right? Yes. So what this model is good for, genetic editing. So you're studying a functional mechanism, high throughput screening because you're getting really consistent, stable, to an extent, tumor organoids, right? I won't touch on microfluidics, but you can also reincorporate native stroma from that dissociated tissue using different microfluidic devices where you're embedding them within the same dome, right? But the same setup. So one paper that used this culture approach that I think is a really great presentation of how it's done and a good application was by this Shen group, I believe, at Columbia. So this was one of the first studies in derivations of bladder cancer organoids. So this was back in 2018, but these were the first bladder cancer organoids in that submerged culture I showed, right? So full dissociation of the tissue, embedding the tumor cells in a matrigel dome, no reconstitution of immune or fibroblast stroma. And because it was the first, they did a really, I would say, a strong job validating this model and then highlighting really the strength of it and doing a high throughput drug screen. So to give you, I guess, a glimpse at what these look like, and to prove that, much like tissue, you can formalin fix, paraffin, embed these organoids and perform histology. You can see here the organoids they derive from primary tissue compared to the primary tumor with H&E, what xenografted tissue looks like, and then what organoids from that xenograft look like. Now, I think it's important whenever you develop these models to perform H&E, but you often see them just presented like this in a figure in a paper with no context. You want to validate these H&E images with a clinical pathologist. You want their assessment of histology, of subtype, of essentially a full clinical pathology assessment. Oftentimes these pictures are just shown, and you're supposed to believe, oh yes, this organoid looks like this primary tumor much more so than this organoid looks like this. No, I think this should be done, but I think it should be done thoughtfully. But more appropriate validation, of course, comes at the genomic and transcriptomic level. So what this group did, and I would say should be done for every organoid model before experiments are performed, is looking at the genome and its concordance with the primary tumor. When you're developing these models, you're dissociating the tumor, you're plating all of these domes. Some will take and some won't, right? The ones that take and the ones you're working with that you pull together on passage, right? What is their concordance? And it's not always one-to-one with the primary tumor. Some drift a bit more, and then I think it's up to you to monitor that and look at that at the time of assay, right? And this is a prime example. So looking at some of these mutations over time, this organoid line isn't bad, right? So the variant allele frequency of the mutations shown here are pretty stable. You're not acquiring or having the outgrowth of any clones with other mutations, right? That's not always the case. Another bladder cancer organoid line, here you see the outgrowth of a increased variable variant allele frequency of KMT2D alteration, and then the loss of others from the primary tumor through organoids at later passage, right? So JAK2 alteration is lost, right? So not only monitoring genotypic concordance with the primary tumor, but tracking it over time and knowing the population you're working with at what passage, right? What that looks like. And not just genomic stability, but phenotype as well. So this paper also looked at the basal and luminal plasticity of these organoid lines. So on the left here, you can see a FOXA1 positive, a strong luminal, less aggressive phenotype that's in the parental tumor and maintained through propagation of the organoids into xenograft and then back into organoids, right? But this SCBO7 organoid line didn't follow suit. So here you see a really luminal lineage of the parental, and then at P0, really, so establishment of these organoids, at 7 days in culture, you start to see evidence of a basal shift, right? Cytokeratin 14 positivity. Out to day 21, that increases, and then by passage 7, you're seeing, purely basal subtype of these organoids. What was interesting, they found that when you graft these organoids into mice, they revert to that luminal subtype, which is interesting. But again, that's lost on extended passage of organoids outside of the mouse. So one, I think it's most important to consider these two things. One, monitoring the phenotype of these organoids when you're working with them. And in general, lower passage is better. If you're going through the effort to establish these lines, if you're doing important work at low passage. And then also some groups do this, the propagation of this tissue material in mice until you're ready to study it, because being in a physiologic system clearly has an impact on the phenotypic drift of some of these populations. And then lastly, I'll speed up a bit. We're going a bit long. But where this reconstituted model is best, the fully dissociated tumor model is in high throughput drug screening. You can see the replicates here. So this is with a MEK inhibitor and an mTOR inhibitor. You're seeing pretty tight groupings with the replicates. And the nice thing about not having your stromal compartments intact, you can do batch analysis of these organoids, for example, here to look at MEK inhibitor impact and show that it's hitting its target, right? You're losing phospho work. So lastly, on the other side of things, a native microenvironment, in general, I would say this is much more like the Wild West, right? I just showed you really clean, consistent ways of validating your reconstituted or, two primary tumor organoids. The more sophisticated you get in keeping the stroma, cellular and acellular stroma intact, the more variance you see in reported work in the validation in those reported works. And I think the derivation of those models, right? Some groups even enzymatically digest to a certain extent these tumors, but not to the point of single cell, but to the point of tissue cluster or cell clusters, right? Others mince, and then some even go to the extent of 1 to 3 millimeter cubed patient-derived tissue fragments, which really isn't an organoid at all. That's more of an explant culture. But the larger the pieces are, the shorter you have in culture. In explant culture, you're talking 24 to 72 hours until you start getting necrosis in the center of that tissue. Whereas when you're talking about a cleanly minced tissue that's not enzymatically dissociated, you can have stable cultures out a few passages, 30 days, a couple months, right? But it requires certain culture conditions, right? So staying away from microfluidics, looking at this ALI culture model, how this needs to be done is in a transwell assay, right? So you have hydrostatic pressure leading to nutrient diffusion through the collagen, but you also have an air interface here. So there's oxygenation of these tissue fragments that keeps the stroma viable and intact for a certain period of time, as we'll see. So here's, I guess, a bit more detailed description. With the transwell, you can add drugs. You could add cellular therapies to this outer chamber, and either through diffusion with hydrostatic pressure, or hopefully, in the case of immune cells migration, those materials can go through a porous membrane at the bottom and travel to the tumor, and you could do perform drug screenings and whatnot, right? This paper, quickly to finish up, used this model and again highlights that it can be done across several types of tumor. There's a lot of work been done here. Like I said, just the validation seems to vary quite a bit between different publications. What they did, I like that it was a short 7-day culture, and then they looked at activation or expansion of native lymphocytes in these ALI cultures. And what they saw was, of course, an expansion, in certain cases, a better expansion with their proprietary nanoparticle than even with checkpoint therapy. This was analyzed via flow cytometry, of course, because you have a mixed culture. Batching in performing assays on these have to be a bit more sophisticated, of course. And then what I'm not showing, they also took the supernatant, if you will, and performed Luminex, right? So there are multiple compartments to study in this model. And then lastly, what wasn't shown in that paper, and again, it kind of corroborates the whole, I think, lack of standardization of these more sophisticated models, is validation. So these are actually images from Kelvin Kuo's group at Stanford, who in 2018 really developed and put out there this ALI, air liquid interface culture technique. These are images of clear cell renal cell carcinoma, ALI organoids. And the important point here is that beyond genotype and phenotype, you also need to track the prevalence of your stromal components, right? So what you can see here compared to fresh tumor, your CD3 positive lymphocytes tend to go away. And this is out to day 30, right? Even at day 7, you see an impact. And the same goes for your fibroblasts, right? So your SMA positive fibroblasts also decline pretty consistently out to day 30. They talked about ways on the immune side, at least, to supplement with IL-2 and stimulate your native TIL population. But again, it's much more preferable to work at low passage, I would say, in these settings. But again, with validation in the ALI, when you have an intact native stromal compartment, it goes beyond genotype and phenotype of the tumor cells. You need to validate the whole 3D structure. So with that simple summary page, but yeah, I think have your assay in mind before you consider each of these models, right? For screenings, for functional mechanism studies, co-culture, straight tumor organoid cultures that are highly reproducible, yet recapitulate the human tumor histology. And then as you want to study more tumor microenvironmental characterizations or even responses to therapies, bring in the native stroma, but understand that there's a bit more cost, much more variability, but that's what we're trying to model in those systems, right? The patient variability we see in the clinic. So with that, thanks to Wilmot Shared Resources, which I didn't put up here, of course, my biobank team, who is terrific, our CCSG P30, and of course, the patients. So. Yeah.
Very comprehensive presentation.
Yeah.
Your experience for the sharp tissue is 1 to 3 cubic. After 30-day culture, you ever passage and those immune cells still can be sustained?
Test the immune cells with what?
In those sharp tissue is not kind of this. So at the very beginning, you still will keep some immune cell. Right?
Yeah.
After 30 days, you ever culture past that and then continue past